So we've met our next speaker earlier this morning, Kangpu Xu, and we learned then that Kangpu is the director of the Laboratory of Preimplantation Genetics, and we even learned what preimplantation genetics was. We can say that Dr. Xu received his training in molecular biology and genetics at the University of Guelph, and then in embryology at the Faculty of Veterinary Medicine in Montreal. He's been at Cornell for some years, where, as I indicated, he's the director of the Laboratory of Preimplantation Genetics. And he's really been instrumental in developing this spectacular new field. And now he's going to tell us about recent advances in preimplantation genetics, diagnosis for cancer susceptibility syndromes. Dr. Xu.
Thank you for the introduction, and thank you for having me here to share our experience. I think I'm going to switch from a population-based to more of a clinical and a family-based individual approach to help prevent cancer. So that's the one. Maybe I should go back. Okay. In the morning, I think I'll try to explain what is a PGD. PGD stands for Preimplantation Genetic Diagnosis, and it's been involved. Perceived in the early days that it is a test tube baby, which is pretty nicely here, and actually it is based on the discovery of DNA. And a few years ago, there's a cross-celebration of first a tested tube baby and also discovery of DNA, which is a part of the nature of a story. So I put them together. We're doing that kind of a work. And so the first case of a PGD is started in 1990, which was published in Nature by a British group, Alan Handyside and his colleagues at Hammersmith Hospital. And at Cornell, we started PGD in 1992. And in the early 1990s, most of our works are efficacy studies, and what are we going to do, which methodology we can take. And basically, there are two basic technology. One is a fluorescent in situ hybridization that can detect an individual on the single cells, or PCR-based that you can amplify DNA, and then you can look into the mutations. And enter in 2000, look in our center, the number doubles from about the 50s to 100. And again, you can see the doubling from 2002. I think this is more or less reflecting the world trend of increasing the use of this technology. If you further analyze what kind of indications, and AS meaning that aneuploidy screening, because we're treating infertile couples and a particular woman of advanced maternal age that are related to oocytes meiotic error, and that we can screening of some of the chromosome, part of a number of the most susceptible the changes of the chromosomes. So as you can see, 60% of our patients are aneuploidy screening. Next one is a single gene defect, including cystic fibrosis, Tay-Sachs, Huntington disease, and now more on cancer predisposition genes. And here also we are doing that for balanced translocation, equally about 13%. And then you have HLA, that HLA typing for the embryo to save a sibling with a cord blood transplantation. And sexing is, for sex-linked disease, very small percentage. And if you move on to 2006, similar percentage, except that we may see an increase in single genes. I think that, although it could be just a variation in year to year, but I think that this is reflected because the Human Genome Project and also understanding and public awareness of existence of PGD, and also the screening test is added on each of the panels. For example, Genzymes are adding on constantly, and New York State's also the screening lab added many genetic disorders. One of those, for example, is a Fragile X, and most of our physicians will add for these infertile patients, added to the list. So therefore, we see increase in the percentages. Of these 180 or 200, 20%, about 40 patients are for single genes. Okay, PGD, I wanted to say that a PGD is really, actually it is truly, it's a team effort. And the physicians at the first line and contact the patient. And Dr. Rosenmax is the director of the Center for Reproductive Medicine. And I think that the fact that a very few ART center is doing PGD, since very few in this country, I think someone has to really believe that this is the part of a technology that leads to the future. So we do have a very committed group of physicians, and then PGD also depends on the embryology lab, that only when you produce viable embryos that it will allow the further testing and resulting healthy pregnancies. Then we have genetic counselors, and we have a small PGD team. And we do have a collaboration with Mark Hughes in Detroit and Valinsky in Chicago. And I'm going to give you a little bit of a background of IVF and embryology. So for PGD to perform, or hypothetical, this morning we discussed a couple that who needs the treatment of in vitro fertilization or assisted reproductive technology, and for PGD. And the woman needs to be stimulated for her ovary, and because in natural cycle you only have 1 or 2 eggs ovulated, and we would like to have a more average in stimulation. Perhaps we'll get 10 or 20 oocytes from one cycle. And then when we get these oocytes at the right time before ovulation occurs, the needle insertion will get eggs, and they will be mixed with the sperm. And in the early 90s, there's one wonderful technology developed. It's called a single sperm intracytoplasma injection, or ICSI in short. You probably would not see a little pipe, a small pour pipette deliver single sperm. This is a single little head there. And that is, first of all, this technology is for infertile male. And secondly, it happened to reduce the risk of sperm contamination for PGD, because during in vitro fertilization or insemination, lots of sperm will attach to a structure called the zonal placenta. And when you biopsy, you may accidentally take a sperm, which is not really the specimen that you wanted to take. So ICSI helps to also PGD. And after fertilization, the embryo will be cultured, and one of the latest development, or cultured for until day 3, or you can culture further to day 5. And that's also the latest development in the last 10 years. The culture media, composition of a culture media has changed, and that much better to support the embryo development, particularly late stages when the requirement is different from, let's say, day 1 to day 3. And when you have the embryos on day 3 or day 5, and they will be put back to uterus through a catheter. And because we are working for in vitro, so we are accessible, or these materials can be accessed in vitro. For example, at a mature egg, we can have, or we can biopsy a little structure called the polar body. This is the products of a first meiotic division. And or after fertilization, this is 2 nuclei. One is from the egg and one is from sperm. And you can take a second polar body biopsy. These polar bodies not going to contribute to the embryo, but will indicate whether it's the embryo carry disease or not carrying disease. However, if you take a polar body, that only reflects the maternal genetic components and not a paternal. So if you are estimating or testing both sites, you have to go after fertilization, for example, day 3, when the embryo reaches 6 to 10 cell stage. And that's by far majority of labs across the world that are taking this approach. Obviously, we cannot take too many cells. That limits our accuracy of the test. So this is the limitation. So one may want to go to day 5 when the embryo reaches 120 cells or 250 cells. Then you can cut chunk of the embryo, which is a trophectoderm, that's not going to contribute to the embryo proper. Here is the inner cell mass. But when you're on day 5, you only have 1 or 2 days left because the embryo usually put it back to the uterus, the synchronized uterus on day 5, and you can postpone to day 6, but then day 7 is really late, or kind of a late that you don't want it to push too late. And that leaves a diagnostic time window is very short. So there's a limit for day 5 transfer. Okay, so that's the whole thing, and the operation of a biopsy is performed here on the inverted microscope, like a nuclear transplantation or injection of genes. You use this micromanipulator, and which is setting on here with 3 pipettes. One is for holding, fixing the embryos. One is for opening the zona pellucida. We need a hole there. And one is taken for the removal of the blastomere, one of the blastomere. I have a short video to show you how that is performed under this microscope. You use acid tyrose as a chemical way to dissolve zona pellucida. And when it's enough, you suck back, so avoid any harmful effect on the embryo, the acid environment. And when the hole is there, you switch a pipette and you approach one of the blastomere, which has a nucleus. May not clear for, but then at this stage of the embryo, the junctional proteins start to express. It's called a compacting. So it's a little harder to really take that cell out, although we use a calcium magnesium free media to facilitate the loosen of these junctional proteins. So it takes a minute or 2, and you carefully remove that cell. And now you have a single cell. And notice that there is a nucleus there. It's healthy, and this cell was subjected to whatever test is FISH-based or PCR-based, and to look at the genes or mutations. And the embryos will keep in the culture dish and in the incubator for another 2 days. Here you have 2 hatching blasts. This is the hole they created. If not, it's still embryo will expand and get out from the shell. And this is one of the biopsy the embryo, or 2 of the biopsy the embryo actually. I don't know which one, but one of those is a healthy girl, 4-year-old now. And these embryos will be placed back. I think I always, this is my favorite slides when I did a postdoc work in Canada, and my colleague did a wonderful experiment that addressed of one concerns, whether you remove one cell, is that safe, and what happens. There's no same kind of experiment done in humans, but in the cow, what she did is separate a 4-cell cow embryo, put it into empty zonal pellucida of four empty ones, and transferred it to 4 separate individual recipients. And now, luckily, it's very lucky that because the cow could be sick and aborted, not because of a quality of a plasmid, but luckily, she got 4 healthy, identical calves. That really demonstrate each individual plasmid is totipotent. They are able to, from single cell at 4-cell stage, develop a whole healthy animal. So, as I said, we do not have the opportunity to do human equivalent study, but in the frozen IVF treatment, we know some of the embryos that after thawing, some of the cells already degenerated, so only part of the embryo survived. That will lead healthy babies. So that supports that if we remove 1 or 2 cells at the embryo early pre-implantation stage, they probably is a safe. It is probably a safe procedure. But also generally ART or IVF is believed to be safe. There are lots of discussions and lots of studies, and many studies. They also raise some questions. Is IVF that may increase or the risk of retinoblastoma? There are also, again, these has a vast, or there are a number of public papers, which I don't have the time to go into detail. But I want to emphasize that the long-term effect on child development is not really clear. That is, at this point, is not clear. Although IVF baby, the oldest, Louise Brown, is 30-some years old now, and she established a family, so you will not see Louise Brown anymore because her name changed. And she has a family, and a long-term, from IVF point of view, is generally safe. But we don't know whether ICSI and culture media exposed for 5 days and removal of cells, is that have an impact? I think we need a study. So overall, as you see, we stimulate the patient, retrieve number of eggs and fertilize and develop in vitro, take one cell and analyze in genetic with different ways, and then transfer back. Hopefully we'll have a healthy baby. At CRMI, in our center, since '93, we have over 200 healthy babies born. And worldwide, I think I haven't seen the latest figure yet, but we'll estimate about 4,000, 5,000 babies, healthy babies. And there is some misdiagnosis, that's another issue. But I think next week in Australia, there's a PGD International Society meeting, which we'll have more data on. So, so we are able to detect these, and on the chromosome level and also single gene level. I'm not intended to read all these, just trying to say that from the beginning of first case diagnosis of cystic fibrosis, now at least more than 150 genetic disorders have been attempted or successfully performed for PGD. This is the continuation of the list, but those blue bars are related to the dominant cancer syndrome. So Dr. Offit from Sloan Kettering Institute published pretty recently, last December, a review article in JAMA that listed 13 disorders that have been attempted or successfully attempted for PGD. So I'm going to talk a little bit of BRCA genes, P53 genes that are EN and retinoblastoma. Those 4 cases are that we have been involved. So first, BRCA genes. I just wanted to every time we started, we always trying to make sure that is the gene sequence. We check the sequence, check the locus, checked out everything that this is the OMI database that we're always based on. And the BRCA genes, I don't think I'm really needed to say that there are lots of experts here, but except the saying that the review or the article published nearly 10 years ago, for women that treated as 10 years of survival rate is about a 50%. I think that has really relevant to our talk of what we see that the demand for PGD for cancer syndrome is because the survival rate has increased. There's more people aware of that, and if survived and relatives and all these, what do you do with this mutation in the family? So that's a question that are really raised and aware of. And we started to approach this as one of the Dr. Rosenberg's fellow, and in late 90s, and during that time, I think testing for BRCA genes is more of a, what should I say, more robust or more accurate. And he thought, why not we do PGD on that? At that time, for mutation analysis, is not as easy as today. And he started to collaborate with a Sloan scientist and then use amplification created a restriction site analysis to detect that particular mutation. We know we use a restriction enzyme to detect other diseases, for example, sickle cell anemia for that particular mutation that can be identified by DDE1. And here, this mutation will not be recognized by the available restriction enzyme. So he used that approach, and generally for PGD, for PCR based, we needed to have enough material from only single cell, which is about 6 picogram of DNA. And we have to amplify most of the time twice and use a nested PCR approach and second PCR, and we get that. And he used that fluorescent label the primer and able to diagnose, distinguish between the mutant allele and the normal allele. And he did lots of testing for, these are the 6 samples with lots of cells. This is a single cell mimic the situation that what we would do in PGD when you only have 1 cell removed. And in particular, we wanted to know a phenomenon called allele dropout. And allele dropout is a phenomenon in particular when you have very lower copy numbers, for example, single cell. And when you amplify the normal allele and you have a mutant allele, the primer may not efficiently anneal at the same time for both. And then you only amplify 1. And if it is a dominant disease and you miss that affected allele, you will definitely have a misdiagnosis. So he tested a lot, and the results are presented in the 2000 annual meeting of the American Society of Reproductive Medicine. And we have not really continued that. The fellow left to finish his work, and we have not really continued the BRCA work, but we did in our center tried a few cases in collaboration with Walensky and Mark Hughes, and we do for BRCA genes. And we also, with a collaboration with the Walensky group, we did a P53 gene, a family of Affymetris disease. And that's mainly the genetic works is done at the RGI and published work and in that article and by Valinsky et al. And there are two cases or two families. Case one is collaborating with our center. It is on p53 gene, exon 5, and the G524A mutation. And this is the family. The profile, female partner is normal, and the male is affected. That's exon 5. And actually, he died, I think, around the time of this attempt. And we did one PGD attempt, 12 eggs retrieved, 12 fertilized, 12 biopsied, and 3 of those selected for transfer, and a healthy baby boy delivered in December 1999. So it must be 8 or 9 years old now. We also did one case for multiple endocrine neoplasia, and again, these are the parameters that we checked at OMI. And when we started work out of these systems, we always check, first of all, the mutations, and this, if you look at the published article from Ann et al., and this is one of the most common mutations on this particular gene, and this couple happened to carry that, and we decided to develop that system, and we amplified and sequenced to make sure there is, in fact, we amplify, the fragments are indeed included in this AC mutation, A2C mutation. And that's the gel, this is the female partner, this is the male, and that happened to be recognized by FOLK1 digestion. This is just regular, normal DNA control. And again, we have a female partner, she's normal, and male carries that mutation. And this couple, well, it's not as lucky as we hoped. And you can see the numbers, instead of a 12 egg retrieval, 12 fertilized, you have 45 of these 3 cycles, and 45 eggs retrieval, only half of those fertilized, and only 13, again, approximately half that can be biopsied, which indicated these embryos, the quality of embryos is not as good, although she is not advanced maternal age, but still you face the situation that the embryo quality is not good. And in one instance, there's no embryo for transfer. In 2 cases that we tried transferred 1, but there's no pregnancy result, that they stopped the treatment. So it's not the happy story, but that's the situation that we may face. Okay, the third case is a retinoblastoma, and that's RB1 genes. It's, again, it is autosomal dominant. The couple, or the man, carries RB mutation on 1351 plus 1 G2A, and it can be recognized by AE2 digestion. Again, we amplified and we sequenced to make sure that we get the right fragments. And we did lots of PCR optimizations, the temperature gradient, and to see the first PCR, second PCR at a different time, and make sure we get the highest amount of the DNA for analysis. This again is the gel that shows the normal pattern. This is heterozygous affected, the father, and that's normal controls. We also label the primers with a fluorescent labeling, and so allow us to do a run on a genetic analyzer. The normal pattern is 109 base pair as here, and heterozygous affected is this band, which is here, and also a large band will not be cut by the enzyme. This is the family, and the father is affected. Here is the fundus. picture of that, and that's the daughter affected, eldest sibling, and after that, and they come to our center, seek that preimplantation genetic diagnosis. This is the first daughter that they have, as you can see, healthy fundus. And the family is very, very grateful, and they agreed to hold a press conference. And as you can see, this is the father, and that's the daughter, affected daughter. And she was successfully treated with a retinoblastoma, but 2 years later she developed a brain tumor, also successfully treated. And that's her sibling. That's the first daughter from PGD, and it's healthy, and actually 2 years or 3 years younger than her sibling. And after a year and a half, they came again, and they have another healthy boy. So obviously, I think at the time of press conference, it's a really touching event. The mother said, I did not realize what, as a mother, the enjoyment, joyful for the mother when she had this little girl, because all the time it was the treating and the emergency room and the hospital visit, and now for these two, it's what a profound relief for individual family. They will not carry that particular mutation. So I think I show these 4 cases that as a part of the 150 that the disorders we're able to do now. It's a remarkable, I think, it's from early 1990s, and less than 20 years we're able to provide, and we're able to help these couples, and not always. We always have some of the, as I see in the MEN couples, that they did not result in pregnancy. But this is a viable option, and the list of 150 is to me is a remarkable. However, if you look at the only update, which I did just 2 days ago, we have 17,000 entries of a Mendelian disorder. They may not really ever need a PGD treatment, but of 150, it's only less than 1% that we're working with. And this is 9 months ago, it's 17,000, now it's 17,700. It's increasing. So what are we facing, and how do we move from now that we are able to help some of the family, but there are definitely more than we can do. So from these, at least I hope that I convinced you that a PGD is a viable reproductive option if the patient have the wish to decrease the impact of cancer syndromes on their next generation. So it is workable. We theoretically, any genetic disorder of known genes or mutation can be candidate for PGD. And just last week, we have one genes that it just worked out at the Johns Hopkins, and nobody else has really published. And we said, well, we'll try, because we can pull on the information online, we can design primers, and we can amplify, and we will detect the mutation. So theoretically, any known that we can work, but it takes weeks or months. But the current strategy and technology is very tedious, and with a limited small groups of researchers, it's very labor intensive, and we can do only a few a year, or a dozen the maximum. So compared to 17,000 or 150 that are already been done, we just would not be able to. So one way is a collaboration, of course, but the other strategy, we have to really do something that make this system more robust and more effective. So take that cystic fibrosis as an example. Again, this is 1992 that the first case published for PGD for CF in New England Journal of Medicine by the same group that published 2 years ago in Nature, Handyside and his colleagues. And now CF is most commonly performed PGD because gene is known, and also many genetic testing is applied, and so that's the best disorder that I work with. However, if you think of can we do 1,000? Right now we have to do one mutation at a time. One mutation, you get a primer, you get an amplifier, you have to optimize temperature, sequence, all these, and can we cope with handle with 1,000 mutation? Just a single disorder is just impossible, or very practically not possible. So we got to do something else, which we can. For example, we can use a linkage marker. This is a couple that come to our center. The father carries most common mutation delta 5-8. The mother is a carrier because they have affected son. Cystic fibrosis affected son. We know that she's a carrier. But the general panel, 97 mutations and all these, will not find any mutation on her. And they wanted to avoid another affected child. So we use the linkage markers and introgenic markers of intron 1 or 2 there, and also we included that one. We test the 4 markers plus the mutation, and we choose 3 more robust ones. And that allow us not only detect the mutation, but also predict which chromosome transmitted from parents. So of the 11 embryos we biopsied, 1 we know for sure that's a normal unaffected because we see both a normal chromosome from mother and normal chromosome from the father. And 3 carriers, and not only we know the carriers, but we know carriers whose mutation, and 3 carriers maternal mutation, which fits the Mendelian pattern very well, and the 3 compound heterozygotes affected, although we don't know which mutation yet, and 1 is inconclusive. As I mentioned, the allele dropout still is a challenge, a problem for us when you work on single cell. So this is at least one step forward to not only detect mutations, that you have these linkage markers. The markers can be used for this couple and also may or may not, depends on informative. But if we have enough marker, that will cover most of the couples. So this is one step forward, allows. And but that's still not enough. You're still working in single genes and in single places. So what we wanted to do is use approach for whole genome amplification. When you get a single cell, you want to amplify in whole genome. And at the beginning of 90s, there's a method published from I think South California, University of South California, and called the PEP. And we tried the single cells. We published a paper in 1994. And we also tried, that methodology is effective, but only coverage is only about 70 to 80%. That is not really high enough to work in a clinical setting. And we also tried DOP to amplify, that is useful for CGH, for example. But for single genes, you have lots of dropouts and problems. And now there seems to be a more of a robust and effective way called multiple displacement amplification, MDA. And we're also working on that, and we are trying to test what is the ratio of allele dropout and how we can check these linkage markers and these. That, if that works, it will allow us to go further and can maybe be able to apply on a chip One of my PhD students is working on that using 10K commercial available Affymetris chip. And this is a publication from German group. This is part of their publication, published paper, figure 3. And this is the diagram from 1P to XQ. As you see that you can detect some aberrations on the copy numbers, which we wanted to see the copy numbers. And one of the things I didn't really touch upon that, we are doing the aneuploidies and we are doing single genes. Last week, for example, one of our patients from Mexico, and they went all the way to New York and did a Huntington disease testing. Happily, they got pregnant, they went home, but they miscarried because trisomy 16, which we did not detect. We will not be able to test. And with this new approach, we hope not only we are able to detect the mutations and very accurately with the linkage markers and the mutations, but also we will detect these aneuploidies, for example, trisomy 16, maybe somewhere you can see the amplification of a 16, the copy numbers. I think the day will come very soon. So I think this, I hope that I convinced you that PGD is a viable option to prevent some cancers, in particular for individual families. But there are lots of technical issues, problems, and also the funding issues, and how we go from here. I wanted to say that at New York State's Department of Health took the lead that in this country, only New York States start to regulate PGD. All the patients that started from New York State, if you want to send a material, you needed to send it to a lab that has the license to do cytogenetics. They have two different sections for regulating cytogenetics or molecular testing. PCR-based, called the molecular testing, and they have to. You can apply either one of those or both. We do have both licenses from that. And from this country, there are only 2 labs so far have the license to do from New York State to do the molecular testing. There are about, I think, 8 or 10 labs to have the license to do cytogenetics. But interestingly, that they start to regulate it, which is a good thing, although in a way I think make us more vigilant, or the procedures are much more effective. But also, I think what all the development from the early 90s and to now is supported by private funding. It's patient-based. I think we really need some support as we now know that PGD is a viable option fighting disease, prevent these disorders. So I happen to add this. I think this is a governance conference. So in 2002, American Scientific American gave the award to Governor Corzine for his leadership, policy leadership. And I have a friend who's doing this rice gene sequencing, so I went there at the New York Academy of Science and met Governor Corzine there. I think he's really, has a sharp mind, and I think we do need not only scientific community, but I think from the legislations and all the government from the state level and the federal level, that we need some leadership here to see, really to support this important research or clinical procedure that help fighting cancer. So with that, I want to thank you.
Great. We have time for 1 or 2 questions.
Screening for the actual mutation, is there a danger that you might actually miss the mutation of interest because of meiotic recombination?
For SNP, we have not. We use SNP just as a linkage marker, not particular SNP. I think for the mutations, let's say P53, whether we want to test that a GG or TT type. I think it's too early to that, but we will use a SNP. I think if we use SNP, we will use many just as a marker to indicate which chromosome that's are transmitted to.
Dan? Hold on just a second, Dan. Yeah.
I wonder if you can review the statistics for another very important genetic disease, and that is sickle cell disease. Has PGD been used for sickle cell disease with great frequency? How does it compare with its use, for example, in cystic fibrosis?
Yeah, that's an interesting question. I think actually sickle cell, PGD for sickle cell, we published in JAMA in 1999. That's the first case live born for sickle cell PGD. And this is, to me, I think it's really complex. We worked out the system, and it's very effective. And but IVF and a PGD is very expensive treatment procedure. As you know, a sickle cell, and it's related to a group, and that relatively has less overall, I think, less resources. And we have not done that many cases, although the procedure is there. And since 1990, we started testing 98. So it's 10 years we've been doing less than 10 couples. Compared to cystic fibrosis, we probably haven't got the exact, the precise number, probably it's 70, 80 couples. So that is, to me, probably it's reflected of the socioeconomic status of that.
Okay.
So that's again, I think we need it.
Okay, thank you. Let's move on.
Thank you.
Thank you, Dr. Xu.